So the reason I mentioned that we've been doing, this is going to be very old, is because there are papers up there that date back to the 1990s, right, and even the 1980s. The problem of multiple sequence alignment is one of the oldest ones in sequence alignment, and unlike doing blast searches and Smith-Waterman and things like that, it is a problem for which we do not have a solution, and we know that it's computationally complicated enough that we're never going to have a solution, okay? So we have some approximations, and towards the end of the talk I'm going to be showing you that the approximations that we've used most of the time have taken us in a particular direction, which is probably not as accurate as we would like. And there are some newer methods that take us in a slightly different direction that certainly work better, but unfortunately are quite a bit slower. Anyway, so there's a big literature here. And I didn't put it in the handouts, but this paper at the very end, the most recent one we have, which is the one that says that the old multiple sequence alignment methods generate alignments that overestimate the amount of positive selection, I think is a really interesting paper. Okay, so the number one thing that you need to know about multiple sequence alignment is that if you're going to do multiple sequence alignment, you already have to know that the sequences are homologous. Multiple sequence alignment methods cannot be used to demonstrate or provide evidence for homology. And I can't say that strongly enough because I've been in lots of seminars where people said, these things are related, here's a multiple sequence alignment. Well, if they're not related, it doesn't make any sense to do a multiple sequence alignment. Multiple sequence alignments don't have any statistics associated with them. They do not provide any evidence for homology. You have to have the homology first. So that is the number one thing to remember. I actually was on a I was an outside reviewer for a student's thesis where she put in an unrelated glutathione transferase into her glutathione transferase alignment, and then she did the alignment and she built the tree, and it all looked perfectly fine, and it's not very surprising that her unrelated guy was really far away from the rest of the tree. But it doesn't make any sense. You can't align things unless they're homologous. Okay? But the alignment programs are not going to tell you that you've done something wrong. They're going to do the alignment. You're going to get a result. So you really have to know this first. So why do we do multiple sequence alignments? And that's getting to what Aaron was just saying. The reason we do multiple sequence alignments is because we need to know where to put the gaps. Okay? If we didn't have gaps, if that was not a way that biology evolution worked, we would not need to worry about multiple sequence alignment. We could just do the alignment, everything would be great, and life would be simple. But as you saw yesterday when we did our Smith-Waterman alignment by hand, I had two different alignments. They had exactly the same score, and you couldn't decide whether or not to put the gap in the G or the G in the gap. Okay? If you add a third sequence or a fourth sequence or a fifth sequence that tell you a little bit more about what's happening around those Gs, it becomes less ambiguous, okay? So we want to figure out where the gaps are. So the problem with multiple sequence alignment is that the most widely used methods we have do something which involves something called progressive alignment, okay? They build successive pairwise alignments, progressive alignment, and the newer ones, like muscle, then revisit things so that they can fix stuff. But the classic methods like Clustal, tea coffee, and some of the other ones like that, basically if you make a mistake early on, and you're not making a mistake on purpose, of course, you're making a mistake because you don't have enough data yet, then that mistake is kind of stuck, and you can't fix it later. So that's what this says. The simple methods basically put the gaps in places early on and they don't get moved. More modern methods have iterative steps that allow you to adjust the gaps. The things we're going to talk about towards the end are more sophisticated methods that involve actually building an evolutionary tree and using that evolutionary tree to guide the alignments. As I said, this is a very old topic. People have been trying to build multiple sequence alignment methods since the late 1980s. And of course, since they're building these methods, they want to know which one's the best. And so there's some real questions as to what are the right tests to use to decide whether or not your multiple sequence alignment is the best one. Okay. So before I go into the actual algorithms, I want to sort of go back and revisit what kind of homology we're talking about when we talk about a multiple sequence alignment. Because we have shifted from this version of homology, which I talked about all day yesterday when we were talking about doing blast searches and statistical significance and stuff like that, to now we're talking about this version of homology. Okay? So our goal is to have a multiple sequence alignment method that really guarantee, well, I won't say guarantee, but does the best possible job of generating alignments where each of the columns in the multiple sequence alignment reflects this one-to-one relationship over evolutionary time. That's our goal. Okay? So this is clearly a much harder problem than this one. So this one, all I care about is unrelated things don't get significant scores, as many related things get significant scores as possible. If I get a few things wrong with the alignment, if some of the gaps are off by 6 or 7 positions, it doesn't matter. The only thing that matters is the E-value. This is a harder problem because now I've got 200 columns. If my protein family is 200 residues long or 400 or 1,000, I have a lot more places that I have to get right. And in order to actually make sure that I'm maintaining this one-to-one relationship. So this is the problem we're going to try to solve. Before I do that, I just want to put this up really quickly. I'm not going to talk about it very much. This is a very brief history that ends back in 1997 of sequence alignment methods. So we've talked a little bit about global similarity. We did an alignment that was a global similarity alignment. It was first described by Needleman-Wunsch. Peter Sellers was a mathematician who had an alignment method which had some nice mathematical properties. When you do a blast search, basically what you're doing is an approximate version of the Smith-Waterman search. One of the big differences between the things that the global alignment methods and local alignment methods is that there's a constraint on the scoring matrix. So the average value of the scoring matrix, we looked at some scoring matrices yesterday. You notice that the diagonal values were all positive. There were some other positive methods off the diagonal for conservative changes, but most of the values were negative. Okay? And you have to have a scoring matrix that has most of the values be negative when you do a local alignment, because if you don't, then making the alignment longer is going to get the score better. So if, sort of statistically on average, the score doesn't get worse if by chance, then alignments basically turn into global alignments. The first rapid method for doing similarity search and alignment was developed actually by John Wilbur and David Lippman in 1983. It was a program called SearchN, and it was the first one to use a lookup-based hashing method for rapidly matching sequences. And that led to David Lippman and I making fast P and fast A, which then led to a statistical thresholds for doing things, which was implemented in BLAST-P. And now we have the current version of BLAST-P, which uses more sophisticated gapping. But everything in this part of the box, which is all the things that we use today for protein sequence searching and for similarity searching in general, are mostly looking for local alignments and have this constraint that the scoring matrix has to have an average value which is less than 0. All right, whoops, wrong way. So I showed you this before, so I'm going to skip over these things. But I want to very quickly show this. So here are some scores for some alignments for things which are sometimes homologous and sometimes not. Okay? So here is hemoglobin versus itself. If you do a global alignment, you might get a score of 725. This is probably using PAM 250 matrix. The local alignment is going to be exactly the same because it's 100% identical to itself. We often sometimes talk about these sequences instead of in terms of similarity, we think of it in terms of distance. Okay, so distance is somewhat the converse of similarity. So if you have lots of similarity, that means you have very little distance. So these guys are 100% identical, so their distance is 0. If you compare beta globin with alpha globin, now you can sort of see just from looking at the scores here that it's about 50% identical, so the distance has gone up. Myoglobin is even farther away. And then we have this leg hemoglobin as a plant globin and so on and so forth. Everything above this row right here are things that are homologous. Okay? But now we go into lysozyme and ribonuclease and cytochrome C. These are all things which are not homologous to globins. If I do a global alignment, then it turns out because I require the alignment to go from beginning to end, the scores become negative. Because these things aren't homologous, it really looks bad. That never happens for a local sequence alignment because remember we had that 0 in the little term where we were taking the maximum of the 4 things. You can't have a local alignment score which is less than 0 because you can always restart at 0. So these numbers simply, the local alignment just gets shorter and shorter and shorter. And distances are things which can also be calculated from global alignments, but not from local alignments, and so they get bigger. So this is an example. All the proteins here that are homologous from beginning to end. They're globally homologous. Here's another example where we're going to be working with calmodulin and other calcium EF-hand binding protein domain proteins where sometimes, so this is obviously globally homologous to itself. It's pretty similar globally to troponin C, but parvalbumin is a protein which has some of these domains, but it has a bunch of other stuff too. So from the local perspective, it has a pretty good score, but from the global perspective, it's quite bad. And here's a protein which has, again, the calcium EF-hand binding domain in one place, but it also has this protease domain. So the local score is not so bad, but the global score is horrible because most of the protein is completely unrelated. In fact, all of these proteins contain a calcium EF-hand binding domain, but most of them have very bad global scores because the domain is in a different sequence context. Then my last example here is this is a sort of one of the first classic examples of a domain shuffled protein where, again, the global score is terrible because there's no global homology, but there are lots of domains which are in different locations in the two proteins which are clearly homologous, and it has a very good local score. So we do local searches, first of all because we understand the statistics, but mostly because we know that our lots of things that we're looking for are not in fact globally homologous. When we do a multiple sequence alignment, now we have a different question. Why would you do a multiple sequence alignment for a set of proteins where they're not globally homologous? Okay. If you've got lots of different domains in different parts of the protein, what sense does it mean to actually try to do a multiple sequence alignment with the entire protein if it's like that? Okay. So as we talk about multiple sequence alignment methods, we'll have methods that basically are looking for global homology or assume global homology, and then we have some other methods that will kind of ignore some parts of it, but I'm going to say that if you're actually doing multiple sequence alignments for some and to address some biological question, it makes a lot more sense to cut out the parts which are in fact homologous. Okay, we already saw this and we already saw this, so I'm going to skip over those. But let's stop here for a second and remind you that if I want to do a rigorous pairwise alignment, then I need to fill in this area. Okay, so the amount of computation I'm going to do is the length of the first sequence times the length of the second sequence. I need to fill in all these circles. So what happens if I want to do a rigorous multiple sequence alignment? Well, let's not be multiple first. Let's just go from 2 to 3. How much more difficult does the problem become? Well, you can imagine that a 3-sequence alignment is really this, and then coming out of the board for the length of the sequence. I go from 2 dimensions to 3 dimensions. Okay? So now, instead of filling in that rectangle, I need to fill in this cube. Okay? And that doesn't actually seem all that unreasonable. And if the only thing we ever wanted to do was align 3 sequences, I'm sure with the computing power we have today, we could do it a lot, all right? Because it's just something which becomes order n's cubed instead of order n squared. The problem is that most people are interested in aligning a lot more than 3 sequences. And in fact, Clustal Omega is very proud of the fact that it can align tens of thousands of sequences because there are tens of thousands, or I guess there are probably now hundreds of thousands of HIV sequences out there. People are very interested in understanding HIV evolution. They want a multiple sequence alignment. So if you do a pairwise alignment, and you've got two sequences that are average protein length of 400 amino acids, then the amount of time it's going to take is going to be proportional to something in the vicinity of 100,000. Okay? If instead of having 2 sequences, you have 10 sequences, then this problem has gotten a lot harder, and it just doesn't matter how much computing power you have, or I mean, also there's problems with the amount of memory that you would have access to. You're not going to be able to do it, because actually, as I said, most people are not interested in doing 10 sequences. They're interested in doing hundreds or thousands or tens of thousands of sequences. Okay. So we don't get to fill in the cube, or we don't get to fill in the 10-dimensional hypercube. We're going to have to find some shortcuts. So and the next slide just shows you what an alignment would look like on this cube. So let's skip that. So what kind of shortcuts do we have? Well, remember that what a multiple sequence alignment does is figure out where to put the gaps. Okay? So the reason that this cube is big is because I need to be able to deal with insertions and deletions. If I jump back real fast to this thing, if I knew that in fact these sequences could be aligned without putting in any gaps, it would be really easy to figure out what the best alignment is. It's either this one, or this one, or this one. It's just going down the diagonal. So the reason I have to worry about the area instead of just looking at a line of nodes, something which is linear, is because I'm going to put in gaps. Okay? So you could imagine if you knew that you could do the alignment without putting in very many gaps, then you wouldn't have to look at everything in the cube. You could just look at this sort of sausage which goes from the beginning of all the sequences down to the end of all the sequences. If you knew that you only required 1 or 2 gaps in order to do it, then the diameter of this sausage would be quite narrow, right? You would just have to look around, and life would be easy. If I knew that I could take 100 sequences and I only needed 10 gaps in order to align all 100 sequences, then I would have this 100-dimensional thing, but I would again be looking at this sausage that was going from one end to the other end of the 100-dimensional hypercube, and just sort of looking in that little tube in there, and life would be good. So for things which are not too distant and where we don't have too many sequences, there are actually some methods that will produce an optimal sequence alignment by using this kind of a optimization. Okay. The problem with, well, so the first problem with multiple sequence alignment is that it's too hard. The second problem with multiple sequence alignment is it's not actually what we want. Okay. So let's talk about that for a little bit. When I go back and I think about doing our circles and filling in our path graph, do the area, or if I do the hypercube and so on and so forth, what I'm doing is calculating a similarity score, which is the sum of lots of mismatch or lots of matches, right? I have all these scoring matrix values that, you know, serine and serine is 2 and tryptophan is 17 and stuff like that. And in a whole bunch of dimensions, I'm adding up all those things to get the best possible score. That kind of calculation for 2 sequences is, it's the sum of the alignment scores for those 2 sequences. If I do it in 3 dimensions, then it's for 3 sequences and so on and so forth. But what we're doing is actually calculating something that's called the sum of pairs score. So that SP right there stands for sum of pairs. The problem with a sum of pairs scores is it is spectacularly not biological. So let's take a look at that here. So here what we're going to do is a multiple sequence alignment of 5 sequences, but we're only going to have one letter in those. It's just one column of those 5 sequences, okay, just to make life easy. So when I go through and calculate the score of a sum of pairs alignment, what I do is what I've done is taken these 5 sequences and I put them at the 5 points of this pentagon. And so every place here where there's a dotted line, what that means is there's been no change. And every place where there's a solid line, that means there has been a change. And so here what I'm doing is talking about the distance in this alignment as opposed to the similarity. And what I say is that In this particular alignment, I've had 1, 2, 3, 4, 5, 6 changes. So I'm going to assign a cost for this particular column of that alignment of 6. Okay? But the thing about this pentagon is that, I guess I could make sort of devil jokes or something like that, but it's not biological. What happened when these sequences changed was something that happened in a tree. Okay? So if I put these sequences on a tree, I could build a tree like this that basically says there was one change. And then these guys all stayed the same, these guys all stayed the same, and the cost is really 1 if I do a tree-based alignment instead of a sum of pairs-based alignment. And the reason I'm talking about this is because almost all the methods that we have, except for some of the recent phylogenetically aware alignments, calculate a sum of pairs score. So they're calculating the wrong thing. Okay. It's probably not as bad as this because most of the time when we're aligning lots of sequences, we don't actually have things which are quite so different at every different place, but we're calculating the wrong thing. Okay. This is what we should be calculating. The problem with calculating this is that building evolutionary trees is actually more computationally complex than doing the alignment. So if you want to build the tree and do the alignment at the same thing, you really are multiplying two things which are spectacularly difficult to do rigorously. But we fortunately have a shortcut, and that's this thing called a star alignment. We can imagine that if you look at this input data, we can say, well, we've got 3 of these and 2 of these, so let's pretend that this guy is the first thing, and we're not exactly evolving, we're kind of exploding, okay? But here we've exploded 3 times and stayed the same, and here we've exploded twice and we've changed, so now the cost of that is 2. Okay. So this kind of an alignment is not biological, but it gives us an answer which is much more reasonable than the sum of pairs alignment. And it turns out that this way of thinking about the evolution of a protein family or a gene family or something like that is also consistent with the way we think about things if we build hidden Markov models or if we build position-specific scoring matrices. Okay, so this isn't right, but it's more right than that is. We'd really like to have this, but we might be pretty happy if we had something like this. Okay. And then to sort of re-emphasize this some more, here we have a sum of pairs alignment for a bunch of sequences, 4 columns. And here we have the star alignment for exactly the same set of data. And what you see is that it's completely different. Although it's not completely different because we're still matching lots of things. But what again happened here is we put the gaps in a different place because multiple alignment is all about figuring out where to put the gaps. So I'm going to just throw this up and pass it by really fast. This is a pretty old paper. There's a method for doing optimal protein sequence alignment, which is practical if you have dozens of sequences that are more than 30% identical. Okay? And the way that this method works is it uses the sausage approach. It turns out that the sequences are not that different. So remember when we did the workshop yesterday afternoon, I had you look at the number of gaps for some of the highest scoring sequences, and it turned out that for the first guy, that was about 60% identical, there was only 1 gap. Okay? So if I have a whole bunch of sequences and they're more than 50% identical and they're protein sequences, I'm not going to have very many gaps, and I can actually align a lot of them because I can use the sausage technique, and life is pretty good. Okay? That's not, in fact, the biological problem that people won't want to solve very much, but it at least means that some things are possible. So this shows you a whole bunch of glutathione transferase sequences, and here is an optimal alignment calculated by this MSA 2.1, and here's the alignment that's calculated by Clustal. And if you look at these things, you can see that they actually look an awful lot alike, but so it seems like they have this gap in the same place over here. But in fact, if you look at the residues which are at the edge of the gap over here and the residues that are at the edge of the gap over there, what you see is that things are not exactly the same. So there has been some kind of disagreement as to where the gaps should go. And I'll tell you, because Clustal W is not an optimal method, it's a progressive method, that almost certainly the case that here Clustal is wrong and the MSA 2.1 is more correct. But the differences aren't very big. Okay, so that's an important thing to keep in the back of your mind is, yes, they're going to be different. Those small but the differences are perhaps, it have to do with where the gaps go. Do those differences matter very much?
When you say more correct, you mean more accurate phylogenetically?
So that's a great question. So what I mean for this is, what the MSA program is trying to do is the best sum of pairs alignment. And what the Clustal program is trying to do is the best sum of pairs alignment. And the alignment here has a better score. If Clustal had been able to figure out to find this alignment, it would have been happier. But because of the way that it works, it missed it. But definitely not more biological because it's a sum of pairs alignment. Okay, so one of the most widely used, and I guess probably today widely complained about multiple sequence alignments is one that was developed by Des Higgins and colleagues. I guess he might have been a graduate student, or maybe he was a postdoc at the time. It's called Clustal and then Clustal W. And it has been cited almost as many times as BLAST, I think. They're maybe busily being neck and neck as to which could be the more important paper for biology. So let's talk about it because it is a prototype. It's of how you do progressive alignment. And it's still widely used. And for lots of problems, it still does a perfectly fine job. But there are better methods. So how does it work? Well, you've got to start a whole bunch of sequences. And basically, the first thing you do is you take all your sequences and you compare all the sequences to all the other sequences. Okay, so if you've got 100 sequences, you're going to take sequence 1, align it with the other 99, and do that 100 times. So you have to do 10,000 alignments. Okay? So having done that, you've calculated lots of pairwise similarity scores. And the next thing you do is this thing called a UPGMA cluster analysis. And so what that means is that the sequences which are most similar to each other come together first, and the next most similar come together next, and so on and so forth. And so then what you do is you take the two most similar sequences, and it says remaining sequences or clusters, and you basically align them. You generate a consensus sequence. You ask whether or not there are more. And so what you're doing, it doesn't show it here, is basically you start out with all these sequences that are kind of on a tree, and you generate a consensus sequence from each of the leaves of the pairs of sequences that are the leaves of the tree. And so that sort of takes you down the tree one step, and then you do that again, and you merge things more and more. So basically you start out with a whole bunch of 2 sequence alignments, and then you end up with a whole bunch of 3 sequence alignments, and then you take the 3 sequence alignments and you align them to each other, and so on and so forth. The problem here is that the structure of the tree that you're using, the order by which you decide which guys are going to be aligned next, is fixed. Okay, so you do not adjust that. You just follow whatever it was that was driving you in the first place. And so the good news is that you could do this and it's pretty straightforward and you get a multiple sequence alignment. The bad news is that if you put a gap in a certain place because you analyzed sequence number 14 against sequence number 20, and it turns out that that gap would have been moved over a couple of places if you had known about sequence number 22, you don't get to do that. Okay, you're just kind of, things get put in and they get stuck. So I don't think anybody in the multiple sequence alignment world, Clustal is always the program that people like to say bad things about. And there are better methods today, but it was the beginning, and it actually, for lots of things, it does pretty well. So let's talk about what that means. Okay. So in order to decide whether or not we have a good multiple sequence alignment program, or to decide whether or not the new program that somebody has proposed is actually a better program, we need to have some kind of benchmarks. Okay. And so this is actually the title of a paper that was published by Julie Thompson that describes something called BaliBase, which is a database of multiple sequence alignments that are largely based on structural data. So it is widely used for deciding whether or not one method is better than another method. And one of the nice things about, so what the paper does for the methods that were available in 1999, it tells you sort of, it gives you some names of programs. And so here's Clustal. Here's a couple of other methods which are long gone. People don't use them anymore. Here's some other methods that people don't use very much anymore. In fact, probably most of these guys, except for Clustal, don't get used very much anymore. But the database that they used of alignments to decide to see which of these methods worked best is still being used. So let's talk about it for a little bit, because I think it's useful to understand how people decide whether or not one method is better than another. They basically have sort of two dimensions of things. They have sets of sequences that are relatively short, sort of average length, and what they call long, which is actually kind of average length. Then we have V1, V2, and V3. So these are sets of sequences which are sort of relatively evenly distributed in evolutionary time, and they have similar lengths. And so we have guys which are less than 25% identical. We have things which are 20 to 40% identical, and we have things that are more than 35% identical. So V1 is the hard guys, and V2 is the intermediate, and V3, you're going to see in just a little bit, are sequences which are very easy to align properly. And then we have these other reference sets, which are sometimes you have a whole bunch of sequences that are more closely related, and then something which is farther. We have things which are different lengths, and we have some sequence families that are more divergent, and some that have some insertions. But we're going to mostly focus on these guys right here. All right, so remember V1, V2, and V3. V1 is hard, V2 is medium, and V3 is easy. And here's, we've got long, medium, and short. And over on the left over here, we have an axis that is telling us how well we're doing. And so what this says is that if you're medium or if you're easy, it doesn't matter whether you're long, medium, or short, you're pretty much always going to get the right answer. But if you're more distantly related, then you're going to do a better job if you've got more data. So that's not a big surprise, but it's sort of reassuring that the data set behaves the way it ought to. Over here, what we're going to do is look at a whole bunch of different methods for doing alignments. So somewhere in here is Clustal X is right there. This is something called PRP, and this is an HMM method, and so on and so forth. And so what you see here is that, with the exception of the HMM method, which nobody uses, everything else over here, all these different alignment methods, and some of them are pattern-based, and some of them are progressive and things like that. Basically, if you're the easy set, then you're pretty much going to be always, more than 95% accurate. If you're the medium set, then you're pretty much going to be always more than 90% accurate. And if you're the hard set, then it matters. Okay, now it turns out that Clustal is going to do better, and PRP is going to do better on this data set, and these other methods don't do quite so well. So for people who are actually doing things with protein families like you might be doing, so one of the things which is sort of not appreciated as I show you this is that these data sets are coming from structural alignments. Okay? So some of these things, remember we talked about yesterday, you know that they're homologous because you've got the structures. If you didn't have the structures, you would not be able to recognize the fact that they're homologous. Okay? So most of the things that you're going to find with BLAST are going to be in this set over here. These guys are things where we manage to do them. We want to really have a hard test so we can discriminate between different methods. But it's not actually something that's going to come up in practice that often because they're very, very distantly related. Some of them we're only recognize that they're related because we have structures. Okay? So the thing that I sort of want you to appreciate about this data set is that yes, this is good at looking to see which method is the best, but maybe it's actually sort of overemphasizing these differences because the data set is actually more challenging than you would encounter in practice. So here are some more things about sum of pair scores. In this case, we're only looking at V1. This is the hardest data set. And again, basically what it says is that Clustal usually does pretty well, and some of the other methods do less well. But we're going to show you some more modern methods that do better than Clustal. So the first one that I'm going to show you is, again, not actually all that much more modern. This paper was published 15 years ago. It's called Tea Coffee. It does something where it's trying to also obviously do a multiple sequence alignment, but it uses a different measure of how well things are working, and it allows you to have, it effectively gives you sort of an iterative way of trying different to put gaps in different places, and it does a better job. Okay, so now we have a whole bunch of different sets of data, and here's Clustal W, and basically it's going to be, you know, Clustal W does this, this, and this, and T-Toffee does better. Okay, and that's as much as I'm going to say about it, because the problem with T-Coffee is that although people feel that it produces better alignments of this particular class of alignments, it's also much slower, and it has a harder time working with very large sets of data. So I'm going to move to this guy, which is one of there are two methods that are, I think, people feel are the most practical and most accurate multiple sequence alignment methods, and muscle is one of them, and the other one is MAFT. I'm not going to talk about that one. So why are these guys better? Well, they're progressive alignment methods, but now instead of doing things with basically doing the alignment or building the tree and then having the tree be fixed, it has an iterative step. So here's an example where muscle is showing that it's better than T-Coffee. So T-Coffee is up here and muscle is down here, and what it's saying is that it's doing a much better job of the N-terminus of this protein, getting everything right and getting this alignment where it is. And you could just sort of see in general, there are a lot of extra gaps in here that somehow are not happening over here. And we believe that that alignment is more correct. So how does it work? Well, it does the all versus all thing. It builds a tree. It builds a multiple sequence alignment. It does then goes through, and basically what happens is this part right over here, where so we build a first tree and we do the multiple sequence alignment, and we build another tree, and then we go through and basically we start trying to rearrange the tree to make things look better. And if they look better, then we take that tree and we redo it and we redo it and redo it, and we do it very quickly. But the important idea here is that the tree is not fixed, that we have this process where we can rearrange things, and that helps us get the gaps in the right place. So here is muscle versus Clustal versus T-Coffee, and we have a different database of benchmarks, but here we have percent identities. So if you are going to be less than 20% identical, then these different methods are going to get things exactly right about half the time, and muscle is going to be better than Clustal, and it's going to be better than T-Coffee, but not much. If you are over here in the more than 70% identical, or even over here, basically you're going to be right 90-some% of the time, and again, it doesn't matter very much. So muscle is better. And the main thing about muscle is it's spectacularly faster, so it's one of the methods that people like to use. Okay, so here are some more things about muscle in BaliBase and so on and so forth, and basically what we're going to see is that the numbers are pretty good, although they're not actually that different from T-Coffee or from Clustal W, but it's what people would be using today. So here we have the optimal alignment, here we have a Clustal alignment, here we have a T-Coffee alignment, here we have the optimal one again, and here we have muscle. Okay? And it basically, there's a whole bunch of stuff in here which is always the same, and then what becomes different is where do the gaps go. Okay. So one of the things that's happening in all these alignments, there are two things. We're doing sum of pairs. We are not thinking about trees. Okay? So there are now some classes of methods for doing multiple sequence alignment that have an explicit phylogenetic model. They want to make sure that the alignments that are being produced, that the positions of the gaps that are being used and stuff like that are actually consistent with something that could happen on a tree. So one of these methods is Prank, which was published by Nick Goldman and his group 7 years ago now. And the thing about Prank is, I'm not sure I should be being taped here. So it likes to put gaps in. Okay, so prank alignments have spectacularly more gaps than other alignments. And those of us who sort of think of alignments in an aesthetic sense are uncomfortable with that. We don't like alignments that have zillions of gaps. It seems like those gaps should be consolidated. But what Goldman and his colleagues argue is that, in fact, if you look at the thing, so just because you have the smallest number of gaps on your alignment does not mean you have the smallest number of events on your evolutionary tree. Okay, so if you're doing things from an evolutionary perspective, it may turn out that more gaps is actually something which is simpler or more parsimonious. And so here's an example of this kind of a thing where here's what actually happened. We put an insertion over in here, and then we have a deletion that's coming over here. And it turns out that if you do a progressive alignment, then you're going to get this particular, so this is the correct answer. And in this case, the progressive alignment is also producing the correct answer. And here's an example where you have a deletion, and this is the correct answer. And progressive alignment is going to produce the correct answer. So life is pretty good. But there are these other times, and they're shown in here, where basically the alignment that you get in evolution is actually, this is the correct answer if you have 2 insertions or 2 deletions. And the alignments that you tend to get from these different methods tend to make those 2 deletions go away. So here we have 2 things happening. We put a G on this branch and we put a C over here. But what's going to happen is I'm going to collapse that gap. And likewise, we have 2 things happening over here. But there are going to be things where I'm going to basically collapse that insertion event as well. Because the methods that we have, at least the sum of pairs methods, they don't know anything about a tree. They don't know that this happened over here and that happened over there. They're just trying to make everything be better as a sum, as a whole. So they tend to make gaps go away. All right, so here is an example where you would have a sum of pairs alignment that was producing something that looks like this. And this would be the same kind of thing, except that it was produced by prank. And there are spectacularly more gaps here. And what Prank is going to argue is that, in fact, yes, there are lots more gaps, but it is evolutionarily much more accurate because, in fact, these gaps are happening in different parts of the tree. So these little symbols over here are pointing out the fact that different things are happening in different places. So they are different events. They're not happening sort of so ancestrally.
So why do they seem so uniform?
Why do they seem?
So nice. Nicely lined up.
So I guess I'm going to say because when you if you have an evolutionary event in one branch of the tree, okay, and then you're going to affect the offspring of that branch, right? But all the other things aren't going to do that, right? So there's going to be lots of stuff which is not affected, and those things are all going to line up, and that's, I think, where you're seeing that uniformity, if that makes sense. Okay. So the last method I want to talk about, I think people today would argue is certainly the one that we wish that we could all use. And it's a program called BaliFi. So I'm not going to be able to explain this very well. And I'm not going to be able to explain this very well, partly because I don't understand it, and partly because it's really complicated, and partly because it's extremely statistical. Okay? So it basically, what Ben Riedelings and his colleagues did was to use sort of modern statistical Bayesian Monte Carlo methods for building trees, and also use those same methods for doing the sequence alignment. And so instead of just sort of deterministically making a particular alignment and then trying to improve it, they're generating a giant distribution of alignments, and they're also generating a giant distribution of trees. And they have costs associated with those trees, which ones are probably better and which ones fit the data worse. They have alignment costs, which ones are more likely and which ones are less likely. And they mix that all together. They do both of those things at the same time. And they get this thing which is called BaliFi, which is a phylogenetically aware multiple sequence alignment. So they know They're building trees, but they're also considering lots of possible trees. They're considering lots of alignments, and they put those things together. And so if this explains it, at least using their words, they would certainly not be my words. But for people who are, so the modern molecular evolution field is very comfortable with this idea that we're going to be doing some kind of posterior sampling of MC space and so on and so forth. So it works. And it is what we want, because what we want is something that makes biological sense. We want to be able to have a tree and build an alignment based on that tree, and then improve the tree based on the alignment, and we're doing that using a statistical strategy that puts those things together very efficiently. Okay, so, but I also say that I teach a much longer version of multiple sequence alignment in a bioinformatics course that I teach, and I was going to try to show how to use BaliPhy to do these alignments and how it would be so much better, and I failed miserably. So either I'm not very patient about reading the documentation, or it's harder, or something. So, but it's what people believe is one of the best methods today. So, does it matter? Well, it turns out that I've been trying to sell the idea that all these alignments kind of look the same. It's just that the gaps are in different places, so maybe it doesn't really matter. And I'm going to say if your sequences are more than 50% identical, it probably doesn't matter very much. Okay? But if your sequences are 30 to 50% identical, or even less than that, then that's another story. And one of the places that these multiple sequence alignments are being used a lot in genome analysis today is to look for evidence of positive selection. Okay, so we have lots of homologous genes. We've got evolutionary trees, so we've got the ensemble data. It's going to tell us about positive selection at different sites and things like that. And what this paper says is that the methods that calculate the, so is everybody clear about positive selection? Let me make sure that, so we have silent changes in sequences, changes that do not change the amino acid sequence. And we have non-silent changes, okay? Substitutions, okay? So where alanine turns into serine or something like that. And there's methods of these evolutionary rate methods that you can use where basically you say, well, I'm looking at the silent rate, and then I'm looking at the non-silent rate, and so I would expect that in general, non-silent substitutions are going to be rarer than silent substitutions because silent substitutions are silent. They're not changing the protein sequence, so they should be able to happily do that because there's not going to be a lot of change. But if you change the protein sequence, for many positions on the protein, you're going to expect that that's not going to be good. It turns out that, I'm kind of making these numbers up, but probably somewhere between 10 and 20% of the positions in a protein actually seem to be like they're under strong selection, so they don't change, where a non-silent mutation is bad. It's actually a large fraction of the protein. It doesn't seem like the protein is actually changing. The silent rate and the non-silent rate are very different from each other. But then sometimes there are places where the non-silent rate, the changes, are actually happening more frequently than the silent rate, which would suggest that there's been positive selection for change, where there's something that's happening where we really need to have that amino acid position change because that's going to allow us to survive. Okay? And the most common examples of those have to do with proteins that are involved in immune surveillance. So people look at antigens on viruses and things like that, and the flu virus is busily avoiding my flu vaccine because it's got positions which change faster than the silent rate, because the ones that have changed are the ones that escape immune surveillance. Okay, so you can go to Ensemble and you can find lots of data about which positions in different genes and the proteins that are coded by those genes are actually under positive selection for change, because that's an exciting question. Because again, it also might tell us something about whether or not these proteins are likely to be acquiring different functions. So basically what this paper says is that our ability to calculate these silent and non-silent rates depends on the multiple sequence alignment. And if you use different multiple sequence aligners, you're going to get different answers. Okay? So, and in fact, the methods that most people use for doing this multiple sequence alignment, these sum of pairs based methods, tend to massively, well, I don't say massively, they overestimate by a factor of maybe 50% to twofold the amount of positive selection that's actually occurring. Okay, so let's take a look at that. So I'm just going to show you these two figures from this paper. Basically, here is, so they have an analysis of, they have 200 data sets from something called the Adaptive Evolution Database. And what they're going to do then is look at a measure of distance between the different sequences or the different predictions from this database. And they're going to do a principal components analysis. And basically what they see is that after they've done their principal components analysis, we have these three clusters. So this is all of our sort of standard pairwise, successive pairwise, or iterative sum of pairs alignment methods. We've got muscle, and we've got MAFT, and we've got Clustal W, and we've got T-coffee, and we've got some other guys. And down over here, we've got Prank, which is the one that likes to put gaps in. And over here, what we have is a whole bunch of different versions, or I would say different versions, but different ways of running based on codons or amino acids and samples and stuff like that, BaliFi. Okay. So the idea is that if you look at the results you get out of this database, the results you get are different depending on how you did the multiple sequence alignment. And over here, what we have is a summary where we're looking at the total number of families out of 200 that are inferred to be under adaptive evolution. Okay? So this is 80. So 200 is over there someplace. Okay? And so we're saying that maybe 40% of the families appear to be under adaptive evolution if we use these methods, but that drops to only 20% of the families if you use these methods. And these methods are all sum of pairs progressive. There's Clustal, there's MAFT, there's muscle, and so on and so forth. So, and the other thing else, that these circles are what we believe the correct answer is. Okay, so there's an enormous difference, basically 80 versus 20 for the progressive alignment, sum of pairs, even more sophisticated methods. Here we have the phylogenetic based methods either using amino acids or codons. And some of these are BaliFi and some of these are Prank. And what you see is that they are also perhaps overestimating how often things happen if they use amino acids. Maybe they're overestimating a little bit less if they're using codon data. But the methods that we've been using that are not phylogenetically aware tend to give much higher estimates for adaptive evolution. And the other thing I need to say is that these are the methods that are being used by Ensemble. Okay, so when Ensemble is telling you that you have all these sites that are under positive selection for change, that is almost certainly an overestimate. Okay, so this is the number one. If they're not homologous, you can't include them in the multiple sequence alignment. So that now gets back to the stuff we were talking about yesterday about domains being homologous, but the entire protein not being homologous, about the possibility that you might have alignment overextension when you've got some homologous domains and then some non-homologous region and then an SH3 domain and stuff like that. So the most important thing you can do, I would say, to get a better alignment is not to use a better program. The most important thing you do to get a better alignment is to make sure that the things that you're aligning are actually homologous. And then all the parts of the thing that you're aligning are actually homologous. Okay.
So in practice, that means you choose the value cut off and then align everything.
Well, I would say if I would today, sort of knowing what I know and knowing what I tried to show you yesterday, it wouldn't just be picking an e-value cutoff and choosing everything. It would be looking at the domain content of the things that were inside that e-value cutoff and trying to decide in those different parts, do they really belong? So for lots of proteins, so what I was showing you yesterday with cortactin was a pathological case. Okay, cortactin is a very mobile domain. SH3 domains are very mobile domains. We've also looked at glutathione transferases and proton transporting ATP synthases and stuff like that. And those guys are well behaved. Okay, so they're not going to trick you. If you just take the entire protein sequence, they always have the same two domains and life is good. But You know, it's important, I guess I would say, and it's really important if you're doing some kind of massive large-scale thing where you haven't actually looked at each of the individual proteins, to be aware of the fact that if it's made up of multiple domains, it's very possible that some of the stuff that you're putting in the alignment should be in the alignment and other parts should not be in the alignment. So you can certainly imagine in that case where, you know, if you just picked all the e-values, you know, even doing things pretty conservatively with the Cortactin example that we were looking at yesterday, you would be putting in a whole bunch of stuff which, you know, one could make the argument was not homologous. And one of the problems with these methods, and it's actually it's sort of a problem downstream, is if I have a whole bunch of sequences that align really well, but then they have some parts where the gaps are crazy, so I do the alignment, I build the evolutionary tree, what ends up happening is that all the good stuff basically has a pretty sensible, consistent distance, and the bad stuff gives me lots of signal that says, I'm different, I'm different, I'm different. And then you end up with an evolutionary tree which is being driven by the part of your data which is the least reliable. Okay, so what you would like is to you know, have a tree which is really driven by the part of the alignment which you actually think is correct. All right? And when people build trees, they now have methods that basically look at the alignments and throw out the bad parts and things like that. But I would say that in general, it would be better if you didn't put the bad parts in the first place.
So you would only align part of the protein?
The part that, if I have a, you know, if I had that, you know, the set of Cortactin domains, I would take the 4 of them that I think, you know, and line those guys all up, and I might put the SH3 domain and stick that on there too and put them together, and I could get a picture of things. Okay? And it would be, I think, a very different picture of things than I would get if I were using the entire protein. So, you know, so I think that's really the issue when you think about doing things on the genome scale, is that when Ensemble produced all the multiple sequence alignments that it produced in order to make all these calculations, they did not look at that. Okay? So in all the cases where life is good and there are a consistent number of domains and things are globally homologous, their numbers are probably great. Okay? And then for all the places where there's something weird going on, there are issues. Okay, other questions? Great.